Y13, another one of these little informative videos. There's the title, Amino Acids to Peptides and Back Again. Just a little reminder first of all, amino acid structure, standard amino acid structure, amino acid. It's called an alpha amino acid because those are on the same carbon. And this bit here, what is termed the R group, is the bit that changes to give you the different amino acids. Now technically there are hundreds of amino acids, but nature, which is where we always focus the discussions about amino acids, there are 22. Most people go, oh, there are 20, but I did a little bit of research earlier. There are 20 what you are called proteinogenic. Yeah, proteinogenic means that the DNA codes them into protein structures, such as enzymes or hormones and all that kind of thing. So the other two have special mechanistic pathways to get them into structures, and I must admit, damn, two amino acids even, I'd not heard of ever. So they were new to me. A little bit of research, never a bad thing. So proteinogenic is 20. And then there's the other two specials. And my cats are right at the door behind me. We had to throw it out just now before we started. It's back. 9 of these 20 are what are termed, for humans this is, that's different for different animals apparently. 9 of these are essential, meaning that your body cannot manufacture them from a different route within your body chemistry. You don't need to know what any of the 20 are, although I recommend you know two, which is glycine and alanine, because they're just two straightforward examples. All the rest, I think you're guaranteed to get given them if you need them. So there are 9 essential, like we're saying. And there are also 3 that are termed semi-essential, which means your body can manufacture them sort of, but it's not terribly good at them, so it's better to have them in your diet. So these have to come from your diet. These often helpfully come from your diet. In reality, all 20 come from your diet, as do the other 2, I suppose. We don't want to get involved in these alternative pathways for getting these involved in structures. That's way too not chemistry for me. Leave that to the biologists for sure. So, just a little reminder about amino acids, and then let's get on to making some peptides. See you in a sec. So, first of all, I want to get you into the way of being able to construct peptides, and then we'll do the deconstruction of them shortly. So, here's the 2 simplest amino acids, if I'm honest. Glycine and alanine. Often known by 3 letters. There's the full name. And condensation reaction kicking out water to form what will be the secondary amide, which is also termed a peptide link. You might like to pause and write that down. You can always do that kind of thing. Stop me and start me and rewind me and make me say it all over again. And when you join those 2 together, you will get what is termed a dipeptide. But when you put glycine with alanine, you could put them together that way and get a dipeptide, or you could do it the other way. I want you to, in just a moment, pause the video and have a go at writing out the 2 structures for glycine and alanine as a dipeptide when they're joined together. So get yourself ready, well, pause me and then get yourself ready if you didn't know that was coming, and I'd like you to pause me, and there are the 2 answers there. There it is with glycine on the left and alanine on the right. Here they're the other way around with alanine on the left, glycine on the right. Now, in reality, if it were in non-neutral, well, if it were in a very acidic conditions, here's your bets there, then if it was very acidic, that would be protonated, like in amino acid chemistry. and if it were in very alkaline conditions, this OH would be losing the H and become a COO minus. So you will get the protonation, deprotonation. But those are the two structures. Now I'm going to get you to have another go, but obviously I need to put the things on the board. I'm going to use amino acids that you don't need to remember the structures for, but you kind of do need to be able to manipulate them, so it's worth having a go with, you know, some of the different ones. I don't remember them either, other than these two. But I will see you after the edit. So, going to get you to have a go at drawing the structure of a tripeptide this time, because 3 amino acids being put together. I'm specifying the order, but obviously you can have, nature would have them not necessarily in this order, depends on what it's doing. Not giving you the structure of glycine, you've already got it. Not giving you the structure of alanine, you've already got it. Here's serine with a CH2OH. Don't want you to do all the different versions. No, that's way too much more than you need to do. Just in that order, glycine to serine to alanine in that order, with the NH2 on that end of the glycine and on this end of the alanine with the carboxylic acid, this end of the carboxylic acid, therefore on the right each time. Put them together, and you're going to pause the video, and when you unpause it, I'll magically put it on the board too. So you're going to pause. So here is the 3 in sequence, 2 water molecules removed, and you can see that was where the alanine came from, because the CH3, there's the serine, and the 2 hydrogens making it glycine. Just the thing about glycine is the only non-optically active, the one without a chiral carbon centre in the middle. All other amino acids are. Now, there is also what happens when you hydrolyse it. Now when you hydrolyse it, you reinstate the NH2 and you break them there and you break it there. Now if you do it with acid, you will get NH3 plus for the glycine. So glycine will do that. This is under acid conditions. If you did it under alkaline hydrolysis, or alkaline I should say, you would have that. You will get a similar structure for each one of those with a different side chain, and it very much depends on whether you've broken up the peptide with acid or whether you've broken up the peptide with alkali. So I'm going to get the board ready for you to have a go at taking them apart, because that's what you can also do is you can hydrolyse them, boiling it in some either acid or alkali. But I need to get the board ready. I'll see you in just a moment. So here is a tripeptide already drawn. There's all the structure all the way through. So what I'm going to ask you to do is, what would be the 3 structures resulting from, what should we have, alkaline, acid, alkaline. Let's go alkaline. Let's do the alkaline hydrolysis products. What would they be? So I'm going to ask you to pause the video, have a go with yourself, and then when you unpause it. So here's the alkaline hydrolysis answers, 3 separated out. Obviously the side chain doesn't get affected in these 2, and here you have to consider what will happen to the NH2, and it will behave the same way as the NH2 at that end of the amino acid. Now if we did acid hydrolysis, we would be protonating, so we would have the carboxylic acid group in each of those positions. That would become NH3+, that would become NH3+, and here you would get 2 NH3 pluses in the acid hydrolysis products. You might like to write that down, which means stopping me and so on. Now, the last thing that we need to look at is what happens if we take it past dipeptides and tripeptides, and the answer is we get to proteins. Little bit about proteins. Cysteine, here the cysteine structure has a really important role to play in part of the structure of proteins. So we're going to do a little bit about protein structures because A-level chemistry doesn't do too much about that in the domain of biology and biochemistry, who are very skilled in these areas. So I'm just going to get the board ready for a little bit about proteins and then we're done. So just finishing up with proteins. Proteins are multitudes of amino acids joined up. You look at the thing, it just goes across the page, loads and loads and loads. So one of the things that you need to remember is that cysteine with the sulfur hydrogen forms a thing called a disulfide link. Now it's not within the chain, it's really between one chain and somewhere else, much further away, where the structure was bent round and the sulfide link then just locks it into position. That comes into play down here. We'll come to that, and I just wanted to remind you that I'd said that already. There are 3 parts to the structure of a protein. The primary structure is just those amino acids in sequence, coded for by the DNA codons, or whatever the proper wording of that is. The secondary structure is formed by hydrogen bonding, and then that's between the carbonyl and the NH parts of adjacent bits of the protein sequence, so they come round and they can do the hydrogen bonding. Then tertiary structure, 3 things cause it. The disulfide links, which comes from the cysteine, so whenever you get cysteine you can start to see those appear. You have instantaneous induced dipole, because of you've got loads of electrons, haven't you, so you're going to get plenty of that going on. And some parts of the structure will have ionic features, and so those will give you what is called a tertiary structure, where the protein is turned into a visible 3-dimensional shape. It's the 3D structure. If they're fibrous, which often means they're in a helix, which is a spiral thing, twisted around, that tends to give you things like muscle fibre. And then you get globular proteins, which tend to be much bigger, and then you get a mixture of the 2 things. You get a sheet structure and you start to see helical structures, and it's quite massively complicated looking structure. Certainly I look at it and go, phew, just a load of twisty shapes and all sorts of things. But these are hormones, enzymes, so when you get the active site, it's all to do with the way that the 3D structure is formed, similarly with hormones. And that's as much as a chemist really needs to know. There we are. There are no follow-up questions. Thank you very much for watching. I'll see you soon. Bye.